In the previous video, we discussed about the invasion of Plasmodium falciparum into RBCs. Now in this video, we will be discussing about molecular biology of Plasmodium falciparum inside the erythrocytes. How Plasmodium functions inside the host erythrocytes. We have an erythrocyte on the right and a Plasmodium falciparum in the blood circulation. From here, this Plasmodium falciparum will get into the host erythrocyte via glidosome and MJ complex formation, which we have already discussed how it invades the RBCs. If you want to watch that video first, you can find the link in the I button or in description. Now we have RBCs with Plasmodium inside. So this RBC is now an infected one, which has capability to block the flow of blood and also changes the behavior of normal RBCs. The infected RBCs show Rosette Formation and Sequestration. Both these phenomena will lead to cytoadherence, which will ultimately occlude the blood vessel. If you see the blood vessel and its circulation, it has got normal RBCs like this. And if we find the infected RBCs with Plasmodium falciparum inside it, then at that time we see some assembly of RBCs in this manner, where the infected cell is at the center and the surrounding cells adhere to it. The structural formation is called the Rosette Formation. And if we see at the walls of blood vessels, some RBCs attached to the endothelial cells of blood vessel, and we call it sequestration. Both rosette as well as sequestration phenomena will cause the occlusion of blood vessel. Now let's see at the molecular level how Plasmodium falciparum performs inside the host cell. First of all, we see this Plasmodium falciparum has got special genes called the VAR genes in its genome, which it expresses once it's inside the host cell. These VAR genes encodes variable adhesion proteins on infected cells, like VAR gene codes for PFEMP1 protein. That's Plasmodium falciparum erythrocyte membrane protein 1. Remember, this PFEMP1 protein has got two domains, DBL and CIDR domain. Both these domains bind to variety of receptors on endothelial cells and also can bind to receptors of normal RBCs. We also see this Plasmodium falciparum expresses KAHRP gene, that's the knob-associated histidine-rich protein gene. This knob-associated protein binds to spectrin, actin, and ankyrin of RBCs. So these are the few important genes of Plasmodium falciparum which encodes these proteins to aid in the cytoadherence. Now let's discuss how the Rosette formation occurs. We see we have an infected RBCs in this blood circulation, and it expresses the PFEMP1 on its surface and also expresses STEVOR protein on its surface. Now we know that normal RBCs expresses diverse of receptors on its surface, and this PFEMP1 shows affinity for multiple RBC receptors through its DBL and CIDR domain. Now looking at this diagram, we see one of the PFEMP1 interacts with CR1 or CD35 receptor of normal RBC. Then we see also this STEVOR binding protein binds to CD36 of normal RBC, while as the other PFEMP1 shows interaction with glycosaminoglycans of normal RBCs. Then we see this STEVOR binding protein also shows affinity for glycophorin C protein on RBC surface. So by showing the following interactions, the normal RBC surround the infected RBC, thus creating a rosette-like shape. So we called it Rosette Formation. So this is how the Rosette Formation takes place inside the blood circulation. This Rosette Formation will also lead to the occlusion of blood vessel. So till now we have seen how this Plasmodium genetically changes the surface properties of infected RBCs and also affects the normal RBCs by Rosette Formation and Sequestration. But there is another important factor that is being changed by this Plasmodium infection, that's the rheological properties. Rheology of blood encompasses the flow properties of the blood, the viscosity of the blood in the circulation. The first normal rheological property of RBCs is the tumbling of RBCs. So RBCs tumble under normal conditions. But when there is an infection in the erythrocyte, the adhesive proteins are lodged to the surface of erythrocytes, which causes the adhesion, that is the cytoadherence. As we have already discussed how infected RBCs adhere to the endothelial cells. If we draw a scale down there showing less shear stress on the left and increasing shear stress on the right, then we figure out under less shear stress, the adhesion enhances in the blood vessels, and if we jump over to increase the shear stress, then at that time the red blood cells show tank treating motion. The cytoadherence caused by the Plasmodium falciparum increases the wall shear stress, thus leading to tank treating motion from normal tumbling. This affects the blood circulation. If you want to know what tank treating motion looks like, then there is a video for that linked in the description. All the above properties and molecular events shows how lethal this falciparum could be inside the cells by expressing its own genes and manipulates the erythrocytes. So this is all about biology of Plasmodium falciparum inside the host erythrocytes. I hope you like the video. If you like it, give it a thumbs up and make sure to subscribe this channel. Thanks.